Good morning, everyone. I'm Annelore Van Nieuwenhove from the Marine Lab of the VUB. Due to the increasing market demand for cephalopods worldwide, many traditional octopus fisheries in the Indian Ocean have expanded very fast in the recent decades and have shifted their focus from local and inland markets to international export. Just as in other regions worldwide, the landings of these octopus fisheries are decreasing and concern over sustainability is raised. To maintain the sustainability of these socio-economic important octopus fisheries, management actions must be taken, such as the establishment of marine protected areas. Insight into connectivity is vital for a correct spacing of marine protected areas. When populations are connected, it means that there is gene flow among those populations or an exchange of individuals among geographically separated subpopulations. Because most of the reef-associated organisms are not very mobile during their adult life stage, connectivity is essentially larval dispersal from one reef to another. When populations are not connected and they don't receive an input of such foreign individuals, local overexploitation might lead to extinction of that population, which can of course be a catastrophe for local communities that rely on fisheries. Because pelagic larvae are extremely difficult to track in the open ocean, population genetics is a useful technique to study connectivity and gene flow patterns. So in order to obtain baseline data for management plans, the present study investigated the diversity of octopodidae in the Western Indian Ocean and connectivity among populations of two commercially imported species, Octopus cyanea and Octopus vulgaris. So how did we do this? We collected arm tips of octopus on landing sites and fish markets at 15 different sites in Tanzania, Kenya, and Madagascar. From those arm tips, the DNA was extracted and a fragment of the mitochondrial cytochrome C oxidase subunit 1 gene, also known as CO1 gene, was amplified and sequenced. CO1 gene is also known as a barcoding gene since it allows scientists to identify the species by comparing the sequence with sequences of many different species in a sequence database. And this results then in an identification summary as output. It's the same principle as scanning the barcode on the product of the grocery store. It's very simple. So besides our own collected tissue samples, we also used 41 CO1 sequences of Octopus vulgaris from different localities worldwide, which are represented by the yellow dots, and these were available on online databases. So besides telling us something about the species, CO1 can also give information on connectivity. How does it work? The more similar the sequences of individuals from different populations are, as you can see on this picture, the higher the gene flow or the connectivity. When the sequences are more different, we say there's high genetic differentiation and therefore low gene flow and low connectivity. So DNA barcoding revealed the presence of 5 different species within our samples, with first records of Callictoctopus luteus and Octopus oliveri in the Western Indian Ocean. For Octopus cyanea, significant genetic differentiation was found among Kanamai, southern Kenya, Tanzania, North and West Madagascar, Southwest Madagascar, and East Madagascar. For Octopus vulgaris, limited connectivity was found among populations from Brazil, populations from Madagascar, and then all the other ones were grouped as one. So what are the take-home messages? There is a higher species diversity of octopodidae in the Western Indian Ocean than previously thought. For Octopus cyanea, we recommend, based on our results, 4 separate management units in the Western Indian Ocean. And for Octopus vulgaris, populations from Brazil and Madagascar were so genetically differentiated that they might be evolving towards speciation and therefore should be considered as separate species in FAO fisheries statistics. Of course, the present study was a bit more complex than the way I presented it here, so people that want more in-depth information on the genetic part or statistical analysis can always talk to me during the breaks. Thank you very much.